I'm very excited to welcome our next speaker, Marna Ericson. Dr. Ericson is the Director of Research at T-Lab. She is affiliated also with the University of Minnesota and the Hormel Institute. Dr. Ericson has done some really amazing work with regard to viewing Bartonella in tissues with confocal microscopy. And today she's going to talk about something even more interesting and novel, which is Bartonella henselae detected in malignant melanoma. Welcome, Marna.
Good afternoon. Can you see my slides?
We cannot yet.
Okay.
Are you sharing your screen?
No, I'm not. I never remember to do that. Hang on. I got it. Are we good now?
Looks great.
Okay, great. Thank you, and thank you all for sticking with us to the end. I'm really excited to have the opportunity to talk at this wonderful day-long seminar. It's worked beautifully, I have to say. And I'm sorry, but all right. I am an employee of T-Lab, but nothing I refer to today will be in any reference to work done there. So I'm going to talk to you today about Bartonella henselae detected in malignant melanoma. It's a preliminary study. And we did publish this paper in March of this year. And I'm going to just walk through it. It's an interesting story, and I think one that we've gotten terribly excited about. It started with our colleague and co-author, Paul Reicheter, when he was out in Oregon, and he's a Mohs surgeon. And he was seeing an inordinate number of patients with malignant melanoma. And he was quite concerned that there was some underlying infection or something going on with these patients. And if you consider that this bacteria is associated with the dermal niche, and also that he had the clinical suspicion that there was an occult infection. And so we set out working together as a team to determine if there was evidence of Bartonella in melanoma. We had published this paper the year before, and this is a multiphoton second harmonic generation image of a patient with Bartonella, and the green dots are the Bartonella and the purple is the collagen in the dermis. And this just solidified what we had actually found in our work. And the little square down in the corner tells me to talk to Dr. Brissett later about she also had some interesting SHG work with Borrelia. And also what we found is when we did culture Bartonella, and we tried it on several different surfaces, it went crazy when we grew it on a collagen surface. That's just an aside, not going to talk too much about that today. Well, here is in this small repertoire or cohort of patients, they were all tested by the group at NC State, and this is before the digital drop PCR method. And 7 of 8 of the patients tested positive by Bartonella, either by serology or PCR, but also take into account these patients, the cultures were only done for 14 days and there was only 1 blood draw and not 3. So it's possible that number 5 could have also tested positive. So malignant melanoma is the 6th most common cancer, as reported by the Center for Disease Control and Prevention. It is caused by a combination of things, sun exposure, genetic factors, and as yet unidentified environmental triggers. And indeed, the survival rate falls to 66% when the disease reaches the lymph nodes and 27% when it reaches distant organs. And what we know about cancer and the microbiome is that to date, and this is probably a low figure, more than 16% of all cancers worldwide have been attributed to infectious agents, such as Helicobacter pylori with gastric cancer, Chlamydia pneumoniae, lung cancer, Salmonella typhi with gallbladder cancer, Streptococcus bovis associated with colorectal cancer. And then we also have the involvement of the murine gut, which was talked about in the last talk. So what we do know is that the association between the bacteria and the tumor microbiome is very complex, and remains poorly understood. So this is a study graphic of the work that we did. So initially, from the patient, we got a biopsy. We also had a blood draw. We did patient enrichment, blood culture, serology, et cetera, which I talked about earlier. And then we did PCR. Also from the tissue, we did PCR as well. That was a nested PCR and from a formalin fixed paraffin embedded section. And then from that, also from that section, we took thick. This is not the best representation of it, but we have about 80 to 100 micron thick tissue, vertical tissue sections. And then we multi-stain those and image those with confocal microscopy. And I'll talk more about all of these in a bit. And just a brief little description of confocal microscopy. And here's just an image of some of the three-dimensional character, and this actually moves, but I don't have the movie on here right now, of skin. And essentially what happens is we take a very thick section as opposed to a 5 micron section. We'll take an 80 or 200, even 200 plus thick section. We'll just optically section it, and then we'll put the loaf of bread back together again. And this is critical, and we want to look at something and like colocalization. We'll use a number of different fluorophores that are isolated from different parts of the visible region. There's literally hundreds of probes and many different lasers and many different other light sources besides this. So the combinations can be overwhelming at times. And this is really important when we want to do verification. So for example, I may want to put DAPI or in this channel, that's a DNA marker. I may put it over here in this channel, and then I'll have a gram-negative probe here and then a Bartonella probe. It's a nice way for us to verify that we actually are seeing what we're seeing, which is demonstrated in this next slide. So this is a full thickness punch biopsy from a non-lesional site on the patient. And this patient was PCR positive to Bartonella, henselae, Vinsonii, and species. And we think it might have been cholerae, but not really sure about that. Patient was always also serology positive and grew in culture. And I can say from this patient, I have many biopsies, and none of them were ever taken from a lesional site, and they always showed up, Bartonella was detected. So here the blue color is the blood vessel, and that's detected with collagen type 4. The green is gram negative, and the red color, which also, when it overlaps with the red, which it should, is Bartonella henselae. So when I initially saw this, my initial reaction was, what did I do wrong? It's all supposed to be here in the endothelial cells and the red blood cells. Well, that's not exactly the case, and that's what we found out. And this is very exciting. So here I just split apart that picture. So the gram-negative bacteria in green, Bartonella, immunoreactive Bartonella henselae in the red. Put those two together, and you get the yellow, which kind of verifies that, yep, we are actually at least picking up gram-negative bacteria. And then I just put it together with a different color blue just to kind of highlight the differences. This has been a really powerful technique for us. What we decided to do is, after looking at those patients, 7 of 8 whom were positive malignant melanoma that were positive for Bartonella, then we had paraffin embedded sections of 2 of them. And both of those patients had been PCR positive for Bartonella henselae San Antonio 2, but they were both seronegative to the panel of Bartonella species antigens that were tested. What we did find is the tissues were PCR positive for Bartonella henselae. They both had BRAF mutations, and I'll show you this data in just a moment. And we did find, this was very exciting, we found that the Bartonella henselae immunoreactivity co-localized with immunoreactive vascular endothelial growth factor C. And we did find it within the lymphatic vessels. So how did we do that? Well, we multi-labeled it in several different staining sets. So the white color is double-stranded DNA picked up by DAPI. The green is immunoreactive Bartonella henselae. The red is vascular endothelial growth factor C. And this lymphatics are picked up by anti-Lyme-1. And you see, if we put both the Bartonella and we project them together, the Bartonella and the DNA probe, indeed, they co-localize. We also found that the VEGF-C co-localize with the Bartonella, and knowing that Bartonella will indeed induce the host to create VEGF-C, this was exciting because we did see this. And this is particularly important since we're looking at a metastatic cancer, wherein blood vessel and lymphatic vessel growth can be an important component for metastases. And then we did find that the Bartonella here in green was found within the magenta colored staining of the lymphatics. Lymphatics are never beautiful. I'll have to say, they kind of are like a loosely knit lacrosse net. All right, so then we got quite interested in what was going on between the Bartonella and the melanoma that we just found in the tissue section. So we did a co-culture model, and we used a melanoma cell line, A375, a metastatic line, and Bartonella henselae from ATCC. And what we found, and again, I'll talk about this in a moment, is we found that Bartonella henselae entered the melanoma cell cytoplasm. And as I reviewed the literature, not the first time we found that. And I think it was Dr. Brightford who told me once he'd never met a cell that Bartonella can't get inside of. So we'll see. That was an aside, but we don't hold them to it. And we also found is that the melanoma cells change shape. And we'll talk about that. And then we measured an increase in VEGF-C, and also interleukin-8, which is a pro-inflammatory cytokine with pro-angiogenic, proliferative, and pro-motility activities. So this is a couple of images from our cultures. This on the upper left-hand corner, this is melanoma alone, no Bartonella. And this is Bartonella in green. And that is now in the mixed culture. And what we did is we actually measured the dendrites, these long processes here. And we found that there was a significant difference, and the Bartonella, the melanoma tended to be shorter and more plump. And then upon closer examination, and then we did this also with a DNA stain, we could see the little yellow dots in here, which are likely Bartonella. And I thought, oh, well, are these on top of, below of, or inside of? So then that led us to the next experiment. And this we did, this is done with super resolution, confocal microscopy and deconvolution, and a bunch of rendering that I was with Ameris that I had help with. And this was exciting because the red dots are Bartonella, the green is the melanoma cells. And excitingly enough, I think some of you may have seen this, but I honestly never get tired of looking at it, of the infiltration of the Bartonella into the melanoma cells, which I am sure you will agree has far-reaching implications. And there we go. All right, so as I said, we did find in the co-cultures, when the Bart and the melanoma were co-cultured, we found elevated VEGF2, my mistake, I misspoke before. And the rate of change was actually much greater when they were in mixed cultures as well. We did find significant initial increases in IL-8 as well. And we were very excited about all of these findings. We do know that Bartonella increases this vascular endothelial growth factor secretion in association with cutaneous vasoproliferative tumor growth. After proteolytic processing, then VEGF receptor 2 is activated, and thus that promotes the angiogenesis and lymphangiogenesis. VEGF is an important melanoma growth factor, and that's what we see in this Bartonella bacilliformis when we see these bacillary angiomatoses. So it's kind of, the story is quite interesting. This paper came out, and this is on the previous work was done by Dr. Conf and DeHayo, and on the Bartonella Auto transporter. And they didn't find that this particular transporter was the one that activated this pathway to drive angiogenesis. And as I said, this came out just soon after our paper, and this was in Nature Communications. Here's a good image, I think, of the lymphatic vessels in the dermis. This is a control patient in white. That is typically how they look. They're kind of closed. And then we have this real unusual looking structure, and you can see all of the processes that have emanated from this Bartonella. And this is a patient that actually had cryptogenic hepatitis and likely had been a Bartonella patient for many years, but this is not uncommon. We also, not with this specific subject, but I want to bring this up because one of the things we're going to do is quantify both lymphatic growth and blood vessel growth in future studies. And we have done that, but not with this particular study set. So what did we find? From this small group of patients, I feel it supports the need for future investigations to determine the extent to which Bartonella are a component of the melanoma pathobiome. Interestingly enough, and I think I have that in a later slide, but I'm not sure, tigecycline has been used as a treatment, and it will indeed delay melanoma growth. In 2020, this seems to be kind of a hot topic recently, and this was in Science, and this group, it's a very large group, they found that they looked at over 1,500 samples and 643 samples and a host of different tumors, and they looked also then at adjacent tissue, and they found that there was over 9,000 bacterial species found in just 7 different tissue types, and it was typically found within the tumor and within the immune cells. And then this paper came out right after our paper did in Nature, and this was really exciting. They looked at, they had, I don't remember how many patients they looked at, but it was a relatively small cohort, but they did a very intensive investigation. And essentially what they found is that peptides that came from the bacteria were being presented by the tumor cells, and they would elicit immune reactivity. This could indeed provide some insight into a mechanism as to how bacteria influence activation of the immune system in response to therapy. So we look forward and we hypothesize that defining the melanoma microbiome is important. It could be a target for preventing initiation or progression, or as was pointed out in perhaps from the last article I just mentioned, it could also influence the immune response. So future studies. Well, more patient samples, that's obvious. We are gonna, we can, because we do this quite readily, we're gonna quantify vasculogenesis and lymphangiogenesis in these samples. We are going to be looking for more pathogens, which are right here. So this is Borrelia in that same sample. And I believe that 4 of the 9 patients, or 4 of the 8, 3 of the 8 patients in this cohort had a history of Lyme disease. We also will try different co-cultures. We can try different cell lines. We can change the timing, like which comes first, the Bartonella. Do you have Bartonella and then you get melanoma? Do you have melanoma and then you get Bartonella? Don't know. And that's another important question to ask. What about expression of different oncogenes? These patients were all BRAF negative, as is very common in melanoma, but we have a whole host of other oncogenes we're interested in looking at. And we are going to be using a mouse xenograft model and knockouts. We have access to this. And then we'll be looking at we're looking at signaling pathways and key components. Now, this is a whole host of things, but the reason I mention that is, oh, I'm sorry, I am one slide ahead of myself. I just wanted to mention this, and I just put this in at the last moment, because you can see this is Bartonella immunoreactive, Bartonella inside the melanoma, excised melanoma lesion. And sometimes we'll find it as the coxoid, the spherical shape. We found it as ribbons, and sometimes a pathetic spirochete, and sometimes a spirochete. This is actually a spirochete in a different patient altogether. And this shows that we found what I'll call this pathetic spirochete in the abdominal skin of a patient with long-standing disease. So the reason I mentioned about doing the signaling, because we'll be doing more work now with our collaborators at the Hormel Institute in Minnesota, and they are quite well, they're very good at cancer signaling and developing new drugs based on some of the work they're doing. They have collaborations at all over Mayo Clinic, MD Anderson, Columbia. And I have to show this because now you're not going to forget. So SPAM, you all know SPAM, spiced ham in a can, was created during World War II to feed the troops because it did not require refrigeration in 1937. In 42, the son of the founder of the Hormel company started the Hormel Research Institute. At that time, it was just a horse barn on the Hormel private estate. And they just actually discovered, they did a lot of lipid research, which is important, of course, for packaging. And they discovered omega-6 and omega-3 fatty acids there. But what I think I find very interesting, and those of you who are too young, go on YouTube and look up the Monty Python SPAM sketch. And it's hysterical. And what happened is, from this funny sketch on TV in 1972 led to the use of the term spam email from this. So now everybody can remember this, and we're very excited about this new collaboration. So as I get to the end here, I just want to thank all the authors and wonderful group of collaborators. And you may have saw this in the earlier slide when I was talking about Dr. Reicheter. This is actually a kangaroo, which is their mascot. I think this is a wolf for NC State. Duluth in the University of Minnesota are bulldogs. And at the main campus at the University of Minnesota, we are the fearsome gophers. This is my reference list, and I'd like to thank you all.
Thank you, Dr. Ericson. That was a very interesting talk.